Welcome. I'm your moderator, Shane Karnick. I'm a Senior Lab Director here at Aliri Bioanalysis. We're excited to have you here for a great discussion centered around artificial intelligence and bioanalysis. And this webinar is co-hosted by Aliri Bioanalysis and BioData Solutions. For anyone who's just learning about Aliri, we are a premier global CRO offering comprehensive bioanalytical, spatial omics, and biomarker solutions that enable and enhance strategic decision-making, strengthen regulatory submissions, and accelerate drug development for our customers. And if you didn't know, we're very proud of our scientific accomplishments, and we're actually just turned 30 years of operations this year. If you don't recognize the name Aliri, it might be because you would recognize one of our legacy brand names, such as Pixent Labs, Emo Biotech, or Tandem Labs, and all these companies are now under the Aliri umbrella. So in this session, we will cover the state of artificial intelligence in the drug development landscape and share real data-backed bioanalytical applications for this powerful tool, including how artificial intelligence in conjunction with genomics, proteomics, and metabolomics analysis can help predict the effectiveness of immunotherapies at an early stage in the drug development process, well before traditional methodologies would allow. So just a reminder, this webinar is being recorded, and after it is completed, we will send that recording out to all the participants. There's also going to be a Q&A box that is open throughout the webinar, so if you have any questions for our panelists, just please drop those questions in there, and we will cover those during the Q&A section at the end. And with that, I just want to introduce our two great speakers today. The first is Stephanie Pastas-Farma, and she is a bioanalytical expert with over 20 years of experience in pharmaceutical, biologics, and hybrid technology. She founded BioData Solutions in 2015 to support bioanalytical drug development through specialized consulting and software offerings. Stephanie has advanced early-stage molecules into potential new drug candidates with bioanalysis support and regulatory compliance planning, as well as led teams of global bioanalytical labs focusing on large molecules and antibody drug conjugates. She holds a PhD and master's in pharmaceutical chemistry from the University of Kansas and a BS in chemistry from St. Mary's College of Notre Dame. We'll also be hearing today from Corinne Ramos. Corinne is our R&D Director at Aliri Bioanalysis. Corinne is a biomarker expert with 25 years of experience in clinical assay, biomarker development, validation, and molecular oncology and imaging in oncology and diagnostics. Corinne received her PhD in molecular biology from the University of Paul Sabatier and an MBA from the University of Johns Hopkins. So with that, I'll pass it to Stephanie, and she will tell us more about AI in the pharma landscape.
Thank you, Shane. I appreciate it. Thank you for also, I'm looking forward to participating in this webinar with Corinne and Aliri. So on the first slide that we see, I wanted to just basically go through what is AI, because we've heard this term often. As you heard from my background, I am a chemist, but I work with AI-enabled tools. And so I wanted to give you a discussion around what are the different types here, because it's a family of different types of software offerings and algorithms. We have machine learning, which we've heard a lot about. There's deep learning, supervised and unsupervised learning. A lot of the predictive models will use ML. We also have natural language processing, NLP. There's also expert systems, which were the first types of AI that go back to the 1960s. There's planning and vision and speech. All of these actually deal with different types of data inputs. Perhaps it's speech to text, or maybe it's some sort of image recognition, but all trying to understand and add some additional utility of taking data in different formats and to providing tools for us to have software enabled to process whatever data we might be doing. So the next slide shows goes into some of what are the applications that we've seen. Across the board, we've seen AI. The biggest impact you'll see highlighted in blue, and the paper is listed below. But we can see that we've seen AI implemented in pharma development from the beginning at research level all the way into sales and marketing. We have a lot of work that we're having, which Corinne will focus on, a lot about, target identification. Some of that helps with understanding biomarkers within a disease state and imaging. But we also even have things like authoring papers. We have Gen AI that's assisting with a lot of this type of SOP work, even down to some AIs that help track interactions of eyeballs on labels to understand what the consumer would draw the consumer to pick out one medicine over another. So it really has led to a variety of different use within our supply chain. On the next slide, we then go into some of the applications that I have been monitoring or seen use within the bioanalytical space, because we are, more highly focused and specialized group. There's a lot of work in bioinformatics, such as Illumina has a primate AI, which helps take some of their NGS data and do some data mining and do some predictive modeling off of some of their sequencing data. But there's also some work in imaging. Report writing has been a big one. As you can imagine, some of the ChatGPT has helped us, and some of the large language modeling, LLMs. Another thing that we have is data review. That's where also my red thread software within BioData that we've brought to the market. But there's also some things like Sapient Sciences. So really trying to work from understanding your data to reporting your data to auditing your data throughout our own workflow in bioanalysis. On the next slide, then we'll go into, well, how is this improved, such as like the clinical landscape, to give you a real world example of what we've seen. There are, first of all, in the first half of this year, each of the 20 top pharmaceutical companies have claimed adoption of some sort of AI in their research and development pipeline. There also have been companies called AI native biotechs, which are companies that have used AI in some sort of part of their drug development process. As you can see on the bottom graph, they've used it to discover targets, maybe to design small molecules, to discover antibodies, discover vaccines. So all across the different types of modalities have used with a lot of understanding of targets and applications, or data mining, looking at past successes and past failures and how that can help design better drugs. And so there are 75 molecules that have gone into the clinic. 67 were in ongoing trials as of the end of 2023. The primary indication has been oncology, but that's not exclusively. And Corinne will talk about her work in oncology specifically, but it can be applied to multiple different therapeutic areas. What is not seen here, and you can actually go and click on this paper, but what they're seeing is a greater success rate in phase 1. I'm conjecturing that this means that we've gotten better molecules through the IND process, through perhaps understanding targets better or better design. So the attrition rates, which is normally like 1 to 5, 1 out of 5 compounds will, will make it into phase 2. That rate has gone up to an 80% of the compounds are going in from phase 1 into phase 2, which is a huge success rate. It is a limited pool, but it is actually very promising that AI has helped us get better drugs designed early on. So it helps us that go, no-go decision and fail or fast, which is what we want. Then on the next slide, we go into, well, what are some of the obstacles that we're running into? Much of what we run into is a data quality or data availability issue. We don't have big data, meaning we don't have 100 pictures of dogs to be able to help, a self-driving car identify that as a dog. We do not want it to drive forward. We just don't have that available. It takes millions and millions of pieces of data to have that. Another thing is finding relevant use cases. Our data has a tendency to be highly biased, all the way down to we just maybe not even have the infrastructure or the framework to actually implement AI appropriately. And so it's not that AI doesn't work, it's just we don't have the capacity to run it correctly. So when we move on to the next slide and we talk about some of the next things of ethical considerations, this is actually a big obstacle that we run into, especially in pharma, I say, because we are a risk-averse people doing a very risky job. What happens, for example, and this is just a hypothetical, what if an AI tool is built for helping diagnose a patient's, maybe oncology, whatever the disease state might be. So the physician interviews the patient, they give all their symptoms. Something is off when they input it. Maybe they weren't given all the symptoms. Maybe something was wrong. We don't know, but the diagnosis is incorrect. And the symptoms could be deadly. And the treatment doesn't work. Who's at fault? Is it the physician? Is it the patient? Is it the AI tool itself, or is it the developer of the tool? We really don't know where the ethical, we don't know where the actual responsibility would lie in this type of case. This doesn't mean that we shouldn't move forward. It just means we need to have guardrails around this to think of the eventuality that this may occur, and so enabling us to avoid this type of consideration, this type of event happening. So when I talk about data biases, what does that really mean? You'll hear the term hallucination. That's just basically when an AI or an algorithm hallucinates. That means you're just given an incorrect or a biased output. How does that come about? You could have data-driven biases. Maybe there's data gaps, or there's biasing of sampling. Maybe you just don't even realize it, but you're already giving it only certain. You're not looking at all different edge cases. The algorithm itself might be biased, and that goes to how you've trained it. You might not have trained it fully, all the different classes and eventualities. It also could be a human bias, meaning social prejudices. There is a huge bias when it comes to some of the AI algorithms as against individuals of color or socioeconomical background, because their data is either not available or it's not included. So it can be a really big thing to consider. So, but just knowledge of this bias is helpful. So when we talk about how are these systems regulated, because we do work in a regulated environment, they're actually treated as a software as a medical device. So, it does have an approval process. There is, the 510(k) process. There's other de novo processes that it must go through. And rather than, most countries are not doing a strict law base, they are doing a soft law approach. That means like a guidance, for example. So the pros of that, actually, rather than being a hard-coded law, is that it can be easily amended, which is helpful with the ever-evolving landscape of AI, and also these systems are very adaptable and constantly changing. However, the con is that they're just voluntary. Since it is a guidance, it is not a required, or there isn't as much of a legal implication if you don't follow these. However, perhaps there are other ways that we are kept in line, but that's the con to this approach. So it gives you flexibility, but doesn't have the legal ramifications that we'd like, perhaps. This just gives you a general, it is a general idea of how these are categorized. We do have a class-based system based on the risk. Class 1 is the least risky, where you may actually be exempt, or you might not have, and the difference between de novo is perhaps there is no precedence. 510(k), there would be a precedent previously, something similar, much like an in vitro device. The riskier ones, where maybe they would be non-exempt, so you would have to go through a 510(k) or a de novo process, but it's less risk as far as the health is concerned. And there's class 3, where you'd have to do pre-market approval, and it is a higher risk approach. You can think of, let's say, some of the imaging that you'd be using to diagnose some disease states that are higher risk, and it could implement, the implications of health, detrimental health, if it's not working appropriately. That might be a higher risk than, let's say, the one I was talking about, imaging on the labeling, right? That would be considered really low risk and maybe not needed to be regulated. This is only for lock systems. Adaptive AI, such as various different MLs, that one is actually an adaptive system, and they are not allowed to be exempt. And that is put into place as of recently, and that's because you don't know how it got from point A to point B, and it's constantly learning and adapting. So they feel like that, right, those types of systems that use adaptive learning like that would need to be constantly monitored and have to automatically go through some sort of approval process. And so, what we run into is really around much of what we work with is in a global environment, right? So all the different leaders are struggling with how to actually governance and oversee AI use in pharma, who has jurisdiction, how do you balance, regulation with innovation? It's always something we grapple with. And what is the government's role in this? And across the actual global industry as well, we always struggle with harmonization across different regions. So that's what we're grappling with. It's no different than other areas of pharma development. It's just that this is moving if, fast, neck-breaking speed with AI development and the things that we're changing. So we're just trying to understand and keep the innovation within our environment, but then how do we actually regulate it? And so what do I mean by AI governance? It literally is with AI especially, you need multiple different stakeholders because it can't just be the software development group governing. You have so many individuals that like compliance, you'd have the actual scientists, they have other different groups that you need to actually have working together. So why do we need it? Because it's rapid pace. Within 6 months, something can be completely different and obsolete. So you have to keep up with that speed of technology and understand how that is constantly changing, perhaps your training or your data sets. It's complex and diverse AI. You have all different types of AI, as I listed. We have everything from computer vision to ML, right? With that level of diversity and the applications are very diverse. How do you govern that? How do you regulate that when all the different use cases are so different? And then also, as I mentioned, determining responsibility. Who, example for that misdiagnosis example, we really need a diverse group helping with governance to understand responsibility and also we need it to be transparent and easily explained to everybody. And that's not always very easy, it's not always easy to understand what maybe I understand and feel like it's obvious might not be obvious to others. So having a cross functional group is really key here so that everyone fully understands the implications of changes and adoption of AI within the workflow. And then there's just, of course, the ethical and social implication when pulling in AI into something where human life is at stake. And then this just goes all the different types of people that may be involved in a governance. Really, it's truly you need to engage all stakeholders. We need to operationalize to have that responsible AI. We want it to be a responsible AI and an ethical AI at its source. So we need to really work within those principles, and we need to evaluate constantly the efficacy of the risk assessments that are applied, the incidences, training. Really, is the algorithm meeting its initial intent? And that doesn't take just a software development group. That takes all the different people involved. Security, cybersecurity, data protection, online safety, because you could just go into ChatGPT and type in information and accidentally leak IP. So we just need people to be properly trained and understand the implications of use of these types of systems. And so in conclusion, we went over some of the regulatory guidance that we have and the proposed guidances, as I mentioned. We have a lot of locked systems guidance out there, but the adaptive is really up in the air right now. We're really trying to go through and understand that. We need to obviously have regulatory guidance that will be adaptable to the changing landscape of AI and have really good communication, not only within our own stakeholder group, but also the end users and the population, the people that we're trying to treat. We could reap a lot of benefits to investment in AI, but we also need to understand that AI currently right now is in a state of, I get a lot of question of how am I going to lose my job? Is it going to take away everything? Right now, AI is only the best success of using AI is when the human and the software work together, the decision maker versus the decision support. AI right now is really in the decision support system. It's not meant to take away the final decision and impact statement because they don't really understand. It can't really understand that that data might be biased. You really need the human in there to understand how it might be biased and to really help assist making decisions, but not take the person out of the decision-making process. So I would like to wrap this up with some, obviously on a positive note, with all of these different ethical consideration and that large stakeholder group, that's a lot of work, right? And we're already very busy. So what is the positive? What are the pros of actually working with AI? Well, as you see up here, this is a recent paper that came out, and there's only 7% of us that are not excited about working with AI at all. There is a great range of individuals that are excited, basically, but 39% are really excited about this. And why? Because we have a lot of data coming at us. And as our molecules get more and more complex, we're getting more and more complex data sets that we just can't fathom getting through all the information that we have. So really handling that more efficiently. Also reduction in human error. Anytime you can hand a repetitive task over to a software, you've gained so much in just reduction of human error and assistance in a repetitive task, which can just be so labor intensive, all the way down to, you know, better quality communication with the stakeholders and developing new services, right? So that's, you know, not everybody is using that right now, but it really is something that we could truly do, and just better all client and wealth and treating of unmet medical disease, you know, needs. So there is a lot of positive to this. It's just we need to go into it with our eyes open of the issues that may come, because it is a very complex system and adoption is not as easy as we had hoped. So with this, I will turn it over to Corinne, who will talk to you about her experience of actually taking AI and helping her with bioinformatics and what she has learned in her experience. So here you go, Corinne.
Thank you very much, Stephanie, and thank you very much for that excellent presentation and for highlighting the value of AI. And building on that, today I will be discussing how advanced technology or advanced analytics can drive better decision-making with a very specific focus on something close to my heart, which is the spatial biomarker. And specifically, I will explore today how AI can enhance early efficacy prediction for lung cancer immunotherapy. But first, with this slide, let's start, you know, like with a bit of history. Pathologists have always aimed to classify disease in order to predict outcome with the ultimate goal of trying to find the best treatment option for the patient. Nowadays, with multi-omics is transforming the predictive science, and with the help of AI and machine learning, we can predict the effect of drug molecule in cell with greater accuracy. On the next slide, you can see that precision medicine requires biomarker. So without, you know, like personalized medicine, the patient will receive any type of therapy. Some will benefit, some of them will not benefit, and a lot of them will have as well, you know, like adverse effect. So only a few portion, you know, like of those patients will get, you know, like benefit from the therapy. With the personalized medicine, there is a focus on trying to find, you know, like the right treatment to the right patient. So with the help, you know, like of the biomarker and specifically with diagnostic biomarker, the patient will receive the therapy that eventually, you know, like they will benefit from. So each patient will, in theory, will benefit, you know, like from the therapy with a personalized medicine strategy. However, this is the theory, and in reality, there is, you know, like some limitation of biomarker. While the biomarker, can you go to the previous slide? While the biomarker can have, we can select actually, you know, like the right population of patients that are positive for a biomarker. Only a small portion of that patient population that are positive for the biomarker will benefit from the treatment. So on the next slide, there is ways of overcoming limitation actually, you know, like to this biomarker. First, you know, like way of overcoming the limitation for biomarker is to train, you know, like better pathologists to better identify the patient that are going actually to respond to the therapy. But we need to see as well, and with the help of AI, biomarker in a much broader way, in the sense that biomarker can interact with each other. And instead of, like looking at one biomarker, we can look, like at an interaction of biomarker or signature biomarker that will help, like to broaden the population that benefit from the therapy. On the next slide, biomarker exist in many different flavors. So you have the biomarker from histology or immunohistochemistry. Histology examine the tissue structure under a microscope to detect abnormality. And immunohistochemistry identify protein in tissue, often used for cancer biomarker. Those biomarkers are really globally implemented. But now, like there is emerging capability, like for example, the transcriptomic that can study RNA to identify active genes in disease like cancer, that has, like a great benefit and has, like a real good moment right now in understanding, like what will be, like the best signature biomarker to predict the therapy. On the next slide, in addition to this, there was, like a breakthrough in terms of targeted therapy in the name of checkpoint inhibitor immunotherapy. So, and you can see, like from the image scan that those checkpoint inhibitor therapy can really improve, and in a very fast way, actually, not only the response to the patient, but as well, like the overall survival. However, do you see as well that there is a limitation at the patient that can benefit from this particular therapy, in the sense that the biomarker that drive, like the response to the particular checkpoint inhibitor therapy needs to be more focused, not only, like on the tumor cell, but as well, like on the tumor microenvironment. And on the next slide, if I want to summarize actually the complexity of immune checkpoint inhibitor and in general immunotherapy, the more complex the therapy, the more complex or the more, like evaluation of biomarker in the different way needs to be accomplished in order, like to be more predictive to the therapy. And if you look specifically at the therapy, not only you need to look at the tumor, but you also need to look, like at how things are arranged around the tumor, like the tumor microenvironment, in order, like to predict, like much better the response to the therapy. And in that sense, and on the next slide, there is, like a groundbreaking specialty right now, which is within, like the spatialomics, spatial biology strategy. And there is a power actually at looking directly with spatialomics in precision medicine and to attack that, like to artificial intelligence in order to increase the accuracy of the therapy. So, for example, in terms of immunotherapy prediction challenge, the predicting success has been always hard because usually the traditional bulk analysis cannot capture the specific interaction between the tumor and the immune cells. And I just showcase, that for this type of therapy, you really need to understand the arrangement of all, like the tumor microenvironment in order to be more predictive. The spatialomics has as well value in the sense that it can map, like the cell interaction, and it uncovers not only the precise localization of immune and tumor cells within tissue that can provide critical context for predicting treatment response. And when you add on top, like of the spatialomic assessment, the artificial intelligence module, you can enhance that prediction, because the AI is going to be able to process the complex spatialomics data to make more accurate, individualized prediction, and to facilitate faster and more effective drug development. And this is an example that you have on the right, where not only you are seeing, like the power of multiplexomics in terms of predicting more accurately the patient that will benefit from an anti-PD-1 drug. But when you build, like on top of this multi-omics capability, special multi-omics capability, when you build, like the AI algorithm on top of it, you will increase accuracy of prediction, and you will increase as well the reliability of your prediction and by interpreting, like multi-omics at the same time. On the next slide. So now, like for cancer, for disease like very complex like cancer, it's very critical to combine, like the AI and the spatial omics in order, like to get into, like precision medicine. So here, like I'm showcasing that integration of multi-omics, genomics, transcriptomic, proteomic, and metabolomic data, and I'm going to follow up with example that can provide, like a much comprehensive insight into biological process, and multiple biological factors across this omic layer can be mined with AI artificial tool in order to predict more accurately the response to the therapy. On the next slide, please. So the process, it's indeed, we are running in a sense a pipeline where we process the image to get a spatial feature. And the speech and the model that we get can classify not only, like the area, tumor versus trauma, but can classify as well cells, tumor, lymphocyte, plasma cells, but we can also classify some objects that are much complex actually onto the image, like for example, the tertiary lymphoid structure, the TLS. And the spatial feature can be categorized in either area region features or cellular distribution features or cellular interaction features, as well as TLS features. And on the next slide, I'm going to start, like giving you some example. So this is an example of resected FFPE lung cancer cohort that those lung cancer were treated with an anti-PD-1 therapy, and we had the response actually, to this particular immune checkpoint inhibitor. So we had, like some responder and some progression disease patient. And what you can see, like by applying spatial omics, in that particular case, we apply spatial proteomics directly on those baseline tissue before receiving the therapy. We can see that we can not only, like multiplex the omics and start, like phenotyping, characterizing just visually what will be, like the different cell immune. See if you go to the next slide, if we apply, like the pipeline that I just described, we'll start, like with image processing, and after that, the nuclear detection. And if you take, like into account that you have, like a cohort of progression disease patient and a cohort of complete response patient, you can start, like segmenting with using some deep learning algorithm. You can start segmenting the cell by using, in that particular case, a deep learning that recognizes the nuclei. And you see, like the performance of this particular deep learning algorithm that are always, like compare, like to the ground truth. And in that sense, it's just, like pathologist evaluation of the cell segmentation. So once you've done, like your nuclear detection and segmentation process, on the next slide, you start, like doing your cell expansion in order to get a nice idea about, like what will be the cell abundance and the cell number within, like the tumor microenvironment that you're looking at. And after that, you can start, like the work of classification on the next slide, where you can make a difference and based, like on the marker that you've been used, like during your multi-omics evaluation, you can start seeing infiltration of T lymphocyte, and this is the case in that particular regards in terms of, like progression disease versus the complete response and the responder. You can see that you get, like much more T cell infiltrated in the tumor microenvironment of the responder. You can go a little bit further in terms, like of phenotyping. On the next slide, the cell, the immune cells. So if you compare again the progression disease patient compared to the complete response patient, in one of them you don't see, like that much infiltration of T cell, and in particular the T cytotoxic cell, which are the cells that are carrying, like in that particular instance, the drug target. And for the complete responder, you are seeing, like much more infiltration of this particular T cytotoxic cells. And you can get, like as well, like a quantification of the drug target within those particular immune cell type that showcase in that particular example that you get, like an immune cell infiltration in the microenvironment of the complete responder, and the percentage of the cell within the drug target, it's over 10%. On the next slide, you can go further in terms of understanding what is the spatial distribution, and also, like the impact of the spatial interaction between those different cells within, like a particular tumor microenvironment, and that can be as well, like predictive of the response to the therapy. And here, what you are seeing here, it's just, like a spatial neighborhood analysis where you can look, like at the distance between, like the immune cells that are carrying the drug target and the cancerous cells. On the next slide, so this is another example of how we can use, like the integration of the multiomics, of the spatial multiomics in that particular cases to answer question again, like about predictivity and try to build, like signature that identify the different, like layer of biological information to really get, like into a signature that can be more predictive actually of the therapy. And in that particular instance, we have used a bioinformatic tool, which is called the Diablo. And the Diablo stands for data integration analysis for biomarker discovery that use, in that particular case, a data variant approach for omics study. But you can use some other, like bioinformatic tool. We can talk about this later. And this allows us actually, like to take the different omics. And in that particular case, I'm going to showcase it's a spatial proteomic, spatial transcriptomic, and spatial metabolic that has been integrated in order to form a signature that could be classified in terms, like of responder and non-responder. So on the next slide, you see the pipeline, the machine learning pipeline, which is used in that case. You start with data preparation. So there is, like really, it's really very important to understand, like the data at the very beginning to also transform the data. When you are working with multiple layers of spatial omics, you need, to transform all the data in order, like to put them on the same level. You need to work as well, like on dimension reduction, because when you are working, like in such a lot of data like this, in particular for the spatial transcriptomic access, you need to understand, like the data at a different level. And after that, you start, like running the Diablo to get, like some variable selection. And I'm going to showcase an example of this. And you can validate, like your signature by looking, like at the treatment response prediction for each, like patient. And finally, like in terms of visualization, you can look, like at the responder, non-responder, and what are the variables that are very important actually within the development of that particular signature, which is going to be customized to every patient. So on the next slide, so this is an outcome of a particular study that we have done on multiple, in that particular case was lung cancer. And we have done, we had, like a cohort of responder, a cohort of non-responder. We run, like spatial proteomics, spatial metabolomics, and spatial transcriptomic on those baseline tissue. And what you get, like after normalizing and putting, like all these different omics on the same level, you can start, like looking at the class separation. So this is exactly what you are looking here. So you get, like the plot, like on the metabolomic side, on the proteomic side, and transcriptomic side. The good thing about running, like spatial biomarker as well and spatial omics as well, that you can really look specifically, like what is the clinical value as well of the different, like region on the tissue. And here you can see that we separate, like the class separation between, like the tumor and the interface. So the class separation showcase that if the ellipse, like are very different, like from each other, that means, that the variable that are being, like visualized are the most, like predictive of a difference between the responder and the non-responder. In that case, the responder are the blue ellipse and the non-responder are the orange ellipse. And the more, like the ellipse are separated, the more, like predictive the variable can be. And here you can see right away that the metabolic being blocked in terms, like of negative as well as the transcriptomic block in the tumor are the more predictive of accuracy of prediction of the response. So on the next slide, this is, like a different way that we can add the data that's showcasing only, the variable that are the most separated within those particular blocks. So if you look, just at the tumor, in that particular case, you are seeing that, for example, the EDO1 gene, in the transcriptomic, it's very well, determinant in terms of separating the non-responder to the responder, and it's also more prominent actually, in the responder cohort, while, the FOX P3, it's the gene, that it's more prominent in the non-responder one, when upregulation can really, if the upregulation of this gene can really determine, the non-responsiveness on that particular cohort for this particular therapy. And finally, when you are at this time, you can start looking, at the signature of all the different omic and by patient. So there are two ways, of validation. You can look at the correlation circle plot that showcasing, the most the different modality, and the closer to each other as also, predictive actually of the response of the non-response to the therapy. And on the right, you will see a heat map that really showcase, each patient we are looking at for each, special different omics layer. And you can start building the right signature for each patient by integrating all this omics modality within the predictivity algorithm. And finally, I just want to terminate actually, this talk by showcasing just, a paper that went out in Nature Methods. It went out, I think, end of August, beginning of September this year, that showcase, the unlocking the power of spatial omics with AI. So this is exactly what I've just showcased to you today, to improve, the predictive power for treatment response. And what we are seeing that AI model, when trained on spatial omics data, they can more accurately predict the treatment response by analyzing the spatial arrangement and molecular state of the cells. And this allows for precise identification of patient subgroup more likely to benefit from specific treatment. And finally, by integrating AI and spatial omics early in drug development, the company can better predict treatment efficacy, reducing late-stage trial failure, and improving clinical success rate. So with that, I'm going to pass on to Shane.
All right. So thank you to Corinne and Stephanie for a very engaging talk today. I do want to say that feel free to continue to submit questions into the Q&A box. We were going to address those. But before we get into the Q&A session, I just wanted to let everybody know that's attending the webinar today that our speakers have offered a free 30 minutes of AI consultation and figuring out how AI can help your drug development programs progress. And so if you'd like to take advantage of that offer, just email them directly and they will set up a time with you to have those discussions. And with that, I'd like to go into the question and answer part and open that up. So we've had several questions come in. So one of the first ones that was submitted is, do you need approval for work that is used in discovery but may become IND enabling? So I guess that's if you have AI research that you've used early on, do you need to get that into approval submission for IND discovery? And so whoever would like to answer that one, please go ahead.
Stephanie, go ahead.
Sure. So I would say that it is currently a gray area. I would say that if it's early discovery and it advances to where it's making decision making, then I would believe that would bring it up in risk, and it would be best to actually go through approval in that point, and also having open discussions with your regulators to perhaps pose that question to them. and do a full risk assessment. So really based on the level of risk, and if it's considered class 2 or class 3, I would personally default to yes, where you would need to go through some of approval. But you're always welcome to have an open discussion with your regulatory group and ask them specifically. So my default would be yes, but it is a gray area at this point.
Yeah, excellent answer. So another question that came in, and this one was just what AI bioinformatic tools are there available in addition to what Corinne covered during her talk?
I can take that
You
one.
want to take that one, Corinne?
So the one I presented was a Diablo tool, but recently actually, like one very nice, like AI tool made the cover of the Nature Biotechnology. This is the cover for October, and it's called, like Stable. So Stable. So those, like have different tool, that will help, like integrate all the different, like multi-omics and special multi-omics in order to be more predictive of a therapy and identify, like multimodal signature that can help, like be more reliable in addressing actually those the specific, like predictivity evaluation. I don't know if Stephanie wants to add something onto it.
So I did see another question, Shane, that says about, it kind of goes with this, is are there AI tools for mass spec data?
Yeah.
That kind of goes in hand. So there are some predictive tools that are available. I personally have not used them myself, but I know like Thermo Fisher has some. I think each one has a various different tools that may go along. I guess it depends on what you're talking about. If you're talking about deep diving into maybe high-res mass data where it can get very complicated, there are some predictive modeling tools that are available. I'm just not 100% sure how those work. Basically on their quality of what they're able to find. But I think that that would actually be a really easy kind of application to talk about some of the data mining strength that AI has. I think that once again, though, it's that conundrum of it is a decision helper, not a decision maker. So you always need to have your oversight of the actual input and output to make sure that it's not hallucinating or giving you some sort of false bias data analysis. But there are some tools available. And I mean, I'm willing to take a call later and discuss it around like how would you kind of figure out how to test it and see how applicable it might be. There was another question as well about like that was directed at me. So I don't know if you're able to see it about like suggest beginning learning more about AI. So I don't know if you want to take that live.
Yeah, if you guys, that would be a good one to jump into. So the question was, what steps would you suggest to begin learning more about AI and how someone could leverage AI to be more efficient in their everyday workflow?
So yeah, so I mean, just to kind of talk about, and Corinne could speak to her experience. When it came to our own delving into software development, algorithm development, AI, even expert systems, we personally at BioData delved into this because we found an area of highly manual, highly exhaustive tasks and tried to figure out if there was a software tool available to help us reduce that so that we would have the ability to increase our throughput and our capabilities and our time savings, which then would basically enable us to do other things. So that is the first evaluation that I would suggest doing as an organization. It would also need senior leadership to be supportive because will take time to evaluate. So the first thing is identification of areas of low efficiency. And then creating a group of stakeholders that should include, in my personal opinion, an end user, someone that's flexible enough and willing enough to play around and fail fast and fail forward. What does that mean? It's a big term, but that means you fail quickly. But you fail forward, meaning you're learning something. It's just like us scientists do every time because you want to continuously move. So sticking in that analysis paralysis is like the worst thing to do. And have like QA, QC, a good group but they're they're agile thinkers and willing to try something new, and also enabling your individuals. But then also understanding risk assessment and clear goals as far as that the first thing that makes an AI die as quickly as possible is that not that it doesn't work, it's just that the people don't have time to actually truly test it. So those are the first steps that I would say is identifying the key area that you're struggling with and making it a pretty simple area. Maybe like it's report writing. That's actually a really easy place to start because you can definitely see the ROI on that. And you can also quickly catch errors if it happens. And then backtracking maybe to more complex workflows, or data auditing is one that we focused on. Stats analysis is another one, something that your group, maybe you're outsourcing and you want to insource, or you want to have a tool. So really identification first, building that team next, but then also having quick risk assessment and goals of how to quickly fail fast, or identify if it's going to work. So that's, and you can reach out to me. We can have a quick call about that as well.
Excellent. Corinne, did you have something you wanted to add on that?
Yeah, in our case, it was a little bit different because we wanted actually to leverage the spatialomic platform and the ton of data that we were generating actually, like for those platforms. So there was a way to be able, like to mine the data. I totally agree with Stephanie that we need, really QA supporting actually those efforts, and also, like a very good team of people able to manipulate those algorithms, because you need to be able to interpret those data. But yes, to us, we were within, the spatialomics evolution, and we really wanted actually to be able to mine the data to get to the accuracy, of algorithm to predict the therapy. But we have, at Aliri, we have a QA group, and we also have a data scientist group, actually, data science group.
Excellent. So another question that came in, I actually find this one very interesting too. I want to hear the answer to, but it is, who is responsible for validating the algorithm? Is it the owner of the company using the tool, or is it the designer of the tools?
So I can start, and maybe Stephanie, you can continue. So we as a vendor, we are responsible actually, of validating, the AI. But we are not responsible of evaluating actually the performance of this AI, and to make sure, that it fits, with the real use or intended use actually, of this AI. So that will be the responsibility of our sponsor.
Okay.
Stephanie wants to add something.
You probably have input.
So no, I think it just really, I echo what you're saying. Yeah, it really does go to vendor management when you're the one using the tool, because it, like you said, it really is up to the vendors. But it also is up to the, the end user to truly do that vendor selection process appropriately and do that risk assessment of how it's being used internally. It goes to that original question, if you're using it in discovery and then it moves into development or high risk sort of diagnostic tools. You just need to have constant iteration also, because that maybe it might shift as well, right, Corinne, of your use of it.
Yeah.
It may shift over time. So the vendor management process might have been appropriate for your intended context of use in the beginning, but it may shift over time. So it's also understanding and keeping your not as static as we have a tendency to have our vendor selection process be, because it really is, the sponsor oversight cannot be understated here. And the sponsor may be a contract lab, and you're not used to being the sponsor in that way, but you're consuming it. So it does need to be more iterative than perhaps in the process of like what we used to do for other vendor selections. You need to have more continuous oversight than previously. It can't be every 2 years.
Excellent. And so we have another question. This one was to Corinne, and it, they thanked you for sharing the lung cancer examples, and they wanted to know, can you confirm whether this applies to other types of cancer disease states as well?
Yeah, I mean, yes, we can definitely work, like on some also cancer. Sure, I mean, there will be, as I said, it's not only, you know, like passing an algorithm, you know, like onto spatial omics data. It's to make sure, you know, that the integrity, you know, like of the spatial omics data for any type, you know, like of, and the sensitivity as well, you know, like of any type of indication. But yes, sure, we can pass along, you know, like on some other, you know, disease, definitely indication.
Excellent. And so, you know, with that, we've actually answered everything that has come into the Q&A thus far, but I do want to say that, you know, we'll continue to submit, if you have a question that comes after the webinar and you'd like to submit it to our panelists, just email Corinne or Stephanie directly and they will get you an answer for that. And with that, you know, I'd like to thank I'd like to thank Stephanie and Corinne for having a very highly engaging talk today. And, you know, I also want to thank everybody for their time that they committed to, you know, attend this webinar and listen to this conversation. And we really hope that, you know, we'll see you at the next webinar that is put on. So thank you very much for your time today. Goodbye.
Thank you.
Thank you.
Yeah, thank you.